Hello everyone. So in this video we are going to discuss about circulatory system lecture 1 of class 8. So our first topic of this lecture is blood groups and blood transfusion. So without wasting time, let's start it. So our topic starts with A, B, AB and O blood groups. Who discovered these blood groups? Firstly, it was discovered by Carl Landsteiner. Carl Landsteiner discovered our blood groups. But glycoproteins on the surface of RBCs are known as antigens, and gamma globulin in the blood plasma are known as antibodies. Therefore, antigens are also known as immunogens. They can be either proteins, lipid, or carbohydrate. But antibodies are also known as immunoglobulin, but they are also certain to glycoproteins. So there are two types of antibodies referred to that A and B are found in human blood plasma. Person with one antigen have natural antibodies in their plasma against other antigen. So that's there. So we have ABO system. Blood group A person which belongs to blood group A have antigen A. If a person belongs to blood group B, it will contain antigen B. If a person belongs to group AB, the person will have antigen A and B both. If a blood group is O, it has no antigen on its surface. So this is our ABO system of blood groups. So we have different human blood groups of their different antigens and antibodies. Suppose we have blood groups of different types. We have AB and O blood group. So let's start with A blood group. If there is A blood group, it will have antigen A on the surface of RBCs and antibody B on the surface of their plasma. The blood group B will have antigen B and antibody of A. The blood group is AB, then it has antigen A and B and antibody none of. It does not contain any antibody. It can be either A or B. If we have blood group O, it will contain none of the antigen. Like A and B does not contain A and B antigen, and antibodies are both A and anti-B. So discuss why do they have different antigen and different antibody. This is because of their clumping. If antigen A of blood group A and antibody from blood group B are not matching, so they are incompatible. Similarly of blood group B also. That's why same. It means when blood from blood group A and B is mixed, clumping or lump formation occur due to antigen and antibody interaction. So, blood transfusion. Person, the one who gives blood is known as donor, and the one who receives blood from the donor is known as recipient. The donor gives blood to recipient. The blood group of donor and that of recipient should be same. If the blood group does not will be same, the clumping reaction occur or the lump formation occur. If A, B of blood group are universal recipient or universal acceptor because they can receive blood group from both blood group A and blood group B. O blood group universal donor because they can donate to blood group A and B, AB only. But they can receive blood group from O only. They cannot receive blood group from A and B. And anticoagulant sodium citrate is added to prevent clotting of blood. If we have suppose clotting in our blood due to some fracture or some trait, the sodium citrate is applied to that. So there are some detectable diseases from blood test. If we have a blood test of glucose level, it is known as diabetes. So what is the cause of diabetes? If pancreas doesn't produce enough insulin or sugar build up in their bloodstream, so diabetes is caused. And therefore hemoglobin estimation is used to detect the disease anemia. Anemia is caused because of the deficiency of iron. And therefore there are certain tests known as RCB count, RBC count, WBC count, PCV, ESR and ELISA test. So that's end of our topic. Lecture 2 will be coming soon of this circulatory system, and lecture 2 of our cell chapter of class 9 will also be coming. So thanks for watching.